Hi, my name is Kris Wood. I'm an associate professor of pharmacology and cancer biology at Duke University, and I'll be presenting today some of our work on using functional genomic tools to interrogate the changes in functional properties of tumors during the course of drug resistance evolution. So our laboratory actually works in two related areas. The first area that we work in is to try to understand how tumor context regulates survival signaling in cancer. Specifically, in this area, we're interested in understanding how things like signaling and metabolic pathways, as well as the structure of organelles like the mitochondria, and even drug treatment exposure, impacts the regulation of survival signaling in cancer cells. Frequently, these studies focus on the BCL2 family of proteins in the mitochondria. The second area of work in our lab focuses on functional tumor evolution. And in this area of the lab, what we're really interested in is understanding how the dependencies in tumor cells change as those tumor cells progress from a treatment-naive state to an on-treatment minimal disease state, and on further to a progressive frank resistant state. And the vignettes that I described today both fall under the umbrella of this second research area in our lab. So today I'm going to describe two stories from the lab that were recently published. The first is a study that involves the concept that drugs can actually exert not only detrimental effects on cancer cells, but also potentially beneficial effects. And that if we can separate out these beneficial and detrimental effects, we can potentiate the activity of anti-cancer therapies. This work was published in Nature Cancer in 2022. The second story I'm going to describe is one that demonstrates that drug therapies can select for clonal variants in tumor cell populations that exhibit exploitable collateral sensitivities. And this is work that was published in Nature Genetics in 2020. Before I go any further, I want to point out that all of the studies I present today, starting with the first story in Nature Cancer, were led by two extraordinary students in my lab, Kevin Lin and Justine Rutter. They were both co-first authors on both of the two papers. And that these studies were also co-led by my colleague and close collaborator, Alex Pouchant, who's at INSERM in the University of Paris. So in the first area, we initiated a study that was motivated by the hypothesis that drugs, and in particular drugs that impact broad cellular processes, might exert not only detrimental effects, but also potentially beneficial effects on the fitness of cancer cells. So to try to understand what these effects might look like, we chose to work with the drug Selinexor, which is an inhibitor of XPO1, a nuclear exportin that is responsible for shuttling protein cargo from the nucleus into the cytoplasm. Selinexor treatment of cancer cells causes an inhibition of XPO1 function and the nuclear sequestration of protein clients. So we hypothesized that the nuclear sequestration of some of these protein clients might actually benefit the fitness levels of cancer cells. So to unpack this, we used two parallel omics approaches. In the top, we used traditional CRISPR-Cas9 based loss of function screens to identify genes that alter the sensitivity of cancer cells to Selinexor treatment. And on the bottom, we used reverse phase protein arrays to understand the changes in signaling pathway activities that are observed in cancer cells following treatment with Selinexor. By combining the results of these two omic analyses, we hypothesized that we could identify pathways and processes that are both activated by Selinexor treatment and whose activation benefits cell fitness. When we did this analysis and we looked at our results, one of the things that stood out was that AKT activation has all the hallmarks of a beneficial effect of Selinexor. CRISPR screens showed that suppression of the PI3K AKT pathway sensitized cells to Selinexor, whereas forced activation, for example, by loss of the tumor suppressor P10, confers resistance to Selinexor. Similarly, we found that through proteomic studies, that treatment of AML cells with Selinexor leads to the activation of the AKT pathway, as judged, for example, by activating phosphorylation marks on AKT at T308 and S473. Together, these lead to the model that suggests that treatment of AML cells with selinexor leads to the activation of AKT signaling, an effect which promotes fitness of these cells. To dig a little bit further, we first showed through flow cytometry studies that AKT activation is really a deterministic effect of AKT activation. So in most, if not all cells treated with selinexor, exhibit an increase in AKT activation. So this is not the selection for minor clones. And if you look across different AML cell lines, we find that AKT activation is a very common observation following selinexor treatment. The activation of AKT following selinexor treatment is an on-target effect associated with XPO1 inhibition. And we know that because if we knock out XPO1 using 2 different guide RNAs, we see AKT activation that phenocopies what we see with the drug. To understand how AKT is being activated by selinexor treatment, we again turn to CRISPR-based screening, this time using flow-based screening approaches, which basically involve first using CRISPR to introduce mutations across all coding genes in the genome, then treating that mutagenized population with selinexor, then flowing those cells through a flow cytometer and capturing those cells that have high or low levels of AKT phosphorylation, where in particular we were interested in gene knockouts that were enriched in the low AKT activation portion of this distribution based on the reasoning that cells that are unable to activate AKT phosphorylation would be enriched in those cells in which key components of the signaling machinery linking selinexor treatment with AKT would have been knocked out. When we looked at the data from these screens, what we found were that while most genes in the genome have no effect or very minor effects on AKT phosphorylation, a handful of genes had dramatic effects. And interestingly, many of those genes are genes that we would associate with the AKT pathway. So for example, AKT1 and 2 itself, the gamma isoform of PI3 kinase, its cognate regulatory subunit PIK3R5, and on the other end of the distribution, PTEN, the negative regulator of PI3 kinase signaling. Now, one of the genes that was most interesting to us was the GPCR P2RY2, which is a known potential activator of AKT signaling that signals through PI3 kinase gamma and PIK3R5 in the GPCR arm of PI3 kinase signaling. Interestingly, when we performed RNA-seq, we found that selinexor treatment not only led to activation of AKT, but also led to the upregulation of P2RY2 expression. And together, these two pieces of data are consistent with the model that the AKT activating function of selinexor treatment might result from the upregulation of P2RY2 that results from drug treatment. We then showed that selinexor-induced P2RY2 activation is not only is both necessary and sufficient for the observed AKT activation. So, for example, if you look across a panel of cell lines, we see consistent P2RY2 upregulation following selinexor treatment. Further, we showed that if you block that upregulation using CRISPR, in this case using doxycycline-inducible shRNAs, we can fully block AKT activation following selinexor treatment. We can phenocopy this effect by using either pertussis toxin, which is a general inhibitor of GPCR signaling, or ARC, which is an inhibitor of P2RY2. And finally, we show that P2RY2 activation is sufficient for AKT activation by showing that if we simply overexpress P2RY2, we can observe AKT activation in cells. To further understand signaling downstream of P2RY2 that's leading to AKT activation, we performed a series of mechanistic studies which revealed that P2RY2-driven AKT activation requires PTEN1 gamma, P101, and also RAS. So for example, if you knock down PI3 kinase gamma or its cognate regulatory subunit PIK3R5, you block AKT activation. And similarly, if you suppress the expression of both KRAS and NRAS, although interestingly not either one on its own, you can similarly block AKT activation following selinexor treatment. And this is consistent with what is known about the role of RAS in signaling downstream of PI3 kinase gamma. From a translational perspective, we were able to observe that inhibiting AKT dramatically sensitizes cells to selinexor treatment by removing this prominent pro-fitness consequence of selinexor. So we see that across a panel of AML cell lines, treatment with structurally and mechanistically distinct AKT inhibitors leads to a frequent sensitization to selinexor. And we find that these results over time lead to a suppression of outgrowth of selinexor-treated AML cell populations in vitro. This suppression of outgrowth and sensitization is associated with the induction of mitochondrial apoptosis, as visualized by caspase and PARP cleavage. And this effect is dependent upon not only the presence of BAX, but also the consequent activation of mitochondrial apoptosis. The combination of AKT inhibition and selinexor is synergistic not only in cell line models, but also in patient-derived samples, where approximately half of a set of roughly 30 samples showed synergy in response to the combination therapy, and another 16 of 32 samples showed additivity, suggesting that the majority of AMLs would be expected to exhibit additivity or synergy in the presence of this combination therapy. Refreshingly, we were relieved and happy to see that normal hematopoietic stem cells do not show the same synergistic response to selinexor plus AKT inhibitor combination therapies, suggesting that there may be a therapeutic window in this combination therapy between cancer cells and normal cells. Lastly, we showed that in a panel of robust AML animal models, the genetically engineered mouse model driven by MLL-AF9, the OCI2 AML cell line xenograft model, and an AML PDX model, that in all three cases, the combination therapy substantially extends overall mouse survival compared to selinexor treatment or AKT inhibition alone. And something that really excited us was that we found that in the particularly aggressive MLL-AF9 genetically engineered mouse model of AML, that the combination of selinexor plus ipatasertib, the AKT inhibitor, not only outperforms the single agent therapies, it outperforms an optimized induction chemotherapy regimen, as well as the combination of selinexor plus chemotherapy. And it outperforms them by a substantial margin. And this was particularly noteworthy to us, not only because induction chemotherapy is the standard of care in young and healthy patients with AML, but also because the selinexor plus ipatasertib combination therapy outperformed induction chemotherapy, even though it was only administered for a few days' time in a manner that reflects the administration of induction chemotherapy. This is noteworthy because we anticipate that in an actual patient, selinexor plus ipatasertib would actually be administered on a chronic basis. Now, to move to the second story that I'm going to share today, in this case, what we were really interested in was looking not at the upfront drug treatment setting, but rather the setting of frank acquired resistance. And specifically what we were interested in was whether cells which develop resistance to a primary therapy might exhibit hypersensitivity to new therapies applied. This concept that the evolutionary changes that lead to resistance to improved fitness in one setting might lead to a decrease in fitness in another setting is known as antagonistic pleiotropy. To understand the antagonistic pleiotropy landscape in AML, what we did is we took OCI-AML2 cells and we performed CRISPR screens either in the presence of DMSO or in the presence of each of a set of 9 different approved and emerging AML chemotherapies and targeted therapies. For the case of each drug, what we documented through these screens were genes and pathways that can either sensitize cells to that drug or confer resistance to that drug. Now, if you look at the results from these 9 screens aggregated together in a circus plot format, what you can do is you can focus in on genes shown in red whose knockout confers sensitivity to each drug, or genes shown in teal whose knockout confers resistance to each drug. Now, what we were most interested in this study were genes whose loss conferred sensitivity to certain drugs, but resistance to other drugs. And the thinking here is that these would be substrates for the development of so-called chemotherapy-induced evolutionary traps, scenarios where treatment of cells with one drug would select for genetic alterations that would also confer increased sensitivity to a second judiciously applied drug. Now, when we look across all 36 pairs of drugs examined in our studies in a pairwise format, what we see is that some drugs share large numbers of co-resistors or co-sensitizers. Those would be those at the top of this graph. Whereas other drugs show large numbers of genes that exhibit antagonistic pleiotropy, meaning that they can confer resistance to one drug but sensitization to another. Perhaps not surprisingly, we found that drugs that work through similar mechanisms of action, like decitabine and azacitidine, share large numbers of common resistors and common sensitizers, like those shown here. Now, more interesting to us, though, were drugs on the other end of the spectrum. Drugs like JQ1, the BRD4 inhibitor, and ABT199 or venetoclax, the BCL2 inhibitor, which showed very different sensitivity modifier landscapes. These drugs were highly enriched for AP genes, scenarios where resistance to one drug conferred hypersensitivity to another. And a great example of this is the cassette of genes shown in this figure panel. As you can appreciate, the loss of certain of these genes causes sensitivity to JQ1, but resistance to ABT199, or the reciprocal effect. Now, to dig in a little deeper, what these data led us to hypothesize was the existence of a PRC2 NSD MYC AP signaling axis that controls sensitivity and resistance to ABT199 and JQ1. Specifically, we hypothesized that NSD23, whose activation would be assumed to activate MYC, we hypothesized that this arm of this cassette could drive resistance to JQ1, but sensitization to ABT199. And that correspondingly, EZH2 and EED, which are negative regulators of MYC, might be seen to confer resistance to 199, but sensitivity to JQ1. To sort of expand this idea, we first manipulated MYC itself, and we showed that knocking down MYC expression indeed confers hypersensitivity to JQ1. This is not surprising, given that JQ1 is thought to act, at least in part, through its suppression of MYC. But that interestingly, knockdown of MYC conferred robust resistance to ABT199. Similarly, overexpression of MYC confers resistance to JQ1 and sensitivity to 199. Now, as evidence that the upstream members of this signaling cassette indeed function through their effects on MYC, we show that NSD2 or 3 knockdown causes a suppression of MYC expression and sensitization to JQ1, whereas knockdown of EED or EZH2 causes an increase in MYC expression and sensitization instead to ABT199. So together, these data validate the concept that MYC activation controlled by NSD2 and 3 and negatively regulated by EZH2 and EED can differentially impact sensitivity to JQ1 and to ABT199. Now, to understand whether this gene cassette driven by MYC is important in naturally evolved resistance and not just functionally contrived settings of resistance, we evolved a panel of 5 AML cell lines to acquired resistance to the BRD4 inhibitor JQ1. When we did this, and then we looked at the Western blotting level, what we observed is that in every setting, cells with acquired resistance to JQ1 showed increased levels of MYC expression. This is, again, consistent with the idea that MYC suppression is critical to the mechanism of action of this drug, and that MYC rebound would be an expected mechanism of resistance. Interestingly, we also found that EZH2 loss in 2 of the 5 models was observed coincident with MYC hyperactivation, suggesting that EZH2 loss might actually be driving resistance in these settings. In a manner that is expected, given that MYC is overactivated in JQ1 resistant cells, we found that these cells were robustly sensitized to the BCL2 inhibitor venetoclax, or ABT199. And finally, we showed that this sensitization to ABT199 was dependent upon the hyperactivation of MYC. Specifically, we showed that if you suppress MYC hyperactivation by partially suppressing MYC levels with shRNA, you could completely reverse the ABT199 hypersensitivity. The MYC's promotion of hypersensitivity to ABT199 is driven by the fact that MYC can cause apoptotic priming in cancer cells. This is not our discovery. This is something that others have shown as well. But what we show is that in the setting of MYC hyperactivation driven by drug therapy, in this case by the treatment with the BRD4 inhibitor JQ1, that this results in an increase of cell priming state as measured by BH3 profiling, and an increase in BIM levels, a pro-apoptotic member of the BCL2 family complex, or family of apoptotic regulators. And in fact, we showed that this BIM upregulation is important for the hypersensitivity to ABT199 that we observe in JQ1 resistant cells, in that knocking down BIM partially reverses the phenotype. Lastly, to understand whether the acquisition of resistance to BRD4 inhibition in vivo leads to hypersensitivity to BCL2 inhibition, we turn to PDX animal models. What we showed using BH3 profiling and the retrieval of cells and functional in vitro assays is that while treatment-naive cells are modestly sensitive to ABT199 therapy, that those cells that emerge following treatment with JQ1 with resistance to JQ1 exhibit hypersensitivity to ABT199 therapy. This validates the idea that the treatment of cells initially with a BRD4 inhibitor primes an evolutionary trap. Consistent with this idea, we showed that scheduling the drug therapies can impact drug sensitivity. Specifically, we showed that a drug therapy that involved first administering JQ1, followed by treatment with ABT199, that's the bottom scheme here, is more effective at suppressing leukemia levels in the body than the reverse treatment schedule, ABT199 followed by JQ1. We see this as read out by the total number of AML cells in the bone marrow, as well as in the number of CD13, CD33 cells in the bone marrow, suggesting the idea that this therapy is leading to frank apoptosis. So in summary, I hope what I've been able to convey to you in this brief talk are 2 stories that demonstrate how functional genomic approaches can be used to better understand how drugs work, and in particular, to understand how to design therapies that make drugs work better. In the first case, by defining the beneficial effects of drug therapies, such that those effects can be neutralized. And in the second case, by defining how drug resistance can select for clonal variants with increased sensitivity to drugs that can be exploited with well-designed evolutionary traps. In addition to the primary authors on this work, I want to acknowledge the other scientists in my lab who are performing studies in one way or another related to these studies to better understand how cancer drugs work and how these drug therapies can change fitness landscapes in cancer cells. And I also want to acknowledge the funding agencies that support our work. With that, thank you for your time today.